Okay. I sense low energy. Tired. Monday, typical Monday. What? No, on Friday. Here in the Marquette area? Maybe up in Negaunee or some of the inland areas. So, well, try to enjoy the weather when you can. I'm guessing that's where some of the people are that are supposed to be here. Maybe they're out enjoying the weather. So, all right. So I'm going to jump around here for a few minutes and then we'll finish off the slide set. So we're almost done, but I just wanted to kind of summarize what we've been talking about. Refresh your memory since it is Monday. It's been, a couple of days off. So this biological nitrogen fixation topic. Can fungi do this? No, exclamation point. Okay, so it is prokaryotic only, and certain key bacteria, and there's some that will never form a relationship with plants and just do it on their own. But we're focusing on the ones that do it with plants, of course, for this class. So when you think about that relationship, it can be very stringent relationship where it's a certain plant species with a certain bacterial species, and it's very tight. There are examples of a more broad host range as well, where the bacteria is actually capable of forming relationships with several different species of plants. So it just depends. And so when you think about the process, initially, the first step. So when I present this information on the exam, for example, I will talk about the different steps, like colonization, like what's going on there. So study it in the different, like step by step of what's happening. So with the colonization, it's this idea where they have to find each other. The plants and the microbe have to find each other. And so, of course, the plant's rooted in place, so they can't go seeking, but they can certainly send out chemicals that can move through the soil. And that's what the bacteria is responding to. And they respond to an increasing concentration gradient. They go toward higher concentrations of that chemical because that's where the root will eventually be found, right? And so then once the bacteria gets there, they have to tell the plant, I'm a friend. I can form a really neat partnership with you. Let's hook up. All right. And so there's this definite chemical interaction that goes on. And then a lot of shifting in gene expression patterns in both partners where they have to kick into this symbiosis phase of their life. And so with the bacteria, they have to attach to the root surface and stay put so they don't get washed away in any rain events, for example. And then usually it's at that step that the root hairs start to curl and branch, and you get that look to the, here's that picture where they're curling. I like to think of it as the root hair is kind of hugging up the bacteria so that they don't wash away, and then the bacteria can make a good physical contact with the root and start carefully degrading the cell wall. They don't want to obliterate it or utterly annihilate it because that would tell the plant it's a pathogen. So they carefully degrade enough of the cell wall that then they can enter into the next phase of the relationship, which unfortunately has the terminology invasion of the root hair. So now once the cell wall is broken down enough, the bacteria will make it past the cell wall, bump up into the cell membrane, bump against it, and then they just kind of push it forward as they migrate down the hair. And so that cell membrane invaginates down the root hair with the microbes pretty much in single file, leading down, moving down the root hair. And then, so that's why they call it the infection thread. It actually looks like a thread under the microscope, so that's why they called it that. So that's like phase 2, colonization, phase 1, invasion, phase 2. And then the setting up camp is the third, where you talk about release of the bacteria. So that's when they've reached their home, the particular plant cells that the vesicle, the plant membrane material will bud off and with the microbes inside. So they're floating around in this vesicle of plant membrane. And they'll set up camp in the different parenchyma cells of the root as the nodule is forming. So they're in the nodule area, and so the plant is busy making that home, doing lots of cell division of its cells there, making that nodule. So then you have the bacteria starting to set up camp in all the cells of the nodule, and then they do start to enlarge. When they get into that phase, they'll enlarge, and then we start calling them bacteroids, and they will promote strong vascular connections to the nodules. So they'll ask the plant to grow the xylem and phloem, essentially get it well infiltrated into the nodule, because they need that in order to give some of the nitrogen to the plant, but also receive the benefit of lots of sugars from the plant. So the bacteria definitely are getting fed. They're getting their nutrient needs met by the plant. And they're, in return, they're fixing that nitrogen that there's not a plant on the face of the earth that we know of that can grab dinitrogen gas and use it. It'd be wonderful if that were the case, but it's just not. And so the bacteria do that very rare reaction using their very special nitrogenase enzyme. So I think we left off right around here, thinking about this idea of getting the vascular connections made, and the plant will definitely give a lot of sugars to the bacteria as they are fixing nitrogen, or both of them. So the bacteria will keep some of the fixed nitrogen, but also export a good portion of it to the plant. So here you're just seeing the final stages the artist drew of getting the vesicles into each cell of the nodule. And then here you're seeing again the visual of a nodule with the naked eye, and then under the microscope you can see pretty much every cell in the nodule has some bacteria. Here's just a close-up, and just trying to show the bacteroids. They're really big. So these large grayish black, those are the bacteria, and you can see the membrane around. So they're quite large in their final phase of this interaction. Okay, so they've looked at this reaction and they've done some calculations, and the upshot is that this is an expensive endeavor. It costs the plant and the bacteria some resources, but the benefit must outweigh the cost, because this is still being done today. It's been going on for millions of years, and we don't see it really going away. So here's the overall reaction, and you don't have to memorize the whole reaction. I do want you to know that dinitrogen, N2, will lead to the ammonia product, NH3. So N2 to NH3. And then the other key thing about this overall equation is that you have 16 ATP. Now you don't have to memorize 16, but know that it's a lot. This is a lot. Okay, so this is an investment in ATP for this to happen. When they do these kinds of calculations, they often frame it from the perspective of how much carbon is the plant giving up in order to maintain this relationship. And for example, soybeans, they've done it for soybean plants, and they say the soybean gives up about 12 grams of carbon just to get one dinitrogen fixed. So again, that's expensive. So it's an investment in carbon and energy by the plant, but the payoff must be worth it, because, like I said, this is still going on. Yes. I don't know. I didn't look that up. So yeah, 1 gram goes to one. What is it? Yeah, the 2 moles. So 1 gram would be. Yeah, that equivalent is good enough. Okay. So yeah, it's expensive, and then you also have to remember. Well, here, let me show a picture of the. It's a multi-subunit enzyme, the dinitrogenase enzyme. Here they're just calling it nitrogenase, it's fine. But you can see it's a multi-subunit protein or enzyme And they're showing over on the right the actual reaction here, this so they know that it's this portion of the enzyme where the nitrogen will dock into the active site, so to speak, and you'll get the reaction to proceed. But you see that there's some other things going on here. They're showing the ATP expenditure here as well. And what they're trying to show in this drawing is that there needs to be a short redox reaction where there's passing of electrons from some of the players in the game here. So, and I wanted to bring your attention to this molecule in the lower left corner, ferredoxin. Okay, so you see the reduced form, RED just means the reduced form of ferredoxin will interact with this portion of the enzyme, the dinitrogenase enzyme. And what they're trying to show here is that this ferredoxin gives off an electron to this other, this one here, this iron. It's in its oxidized state. So what they're trying to show is this ferredoxin and this iron react, and the ferredoxin gives an electron to the iron, and then the iron becomes reduced. And then the iron moves and interacts with the molybdenum iron complex, and they have an exchange going on there too. So the upshot is it's an electron redox reaction, electrons getting passed in order to allow for this reaction to fully occur. And ferredoxin in particular, I want you to keep in mind, because ferredoxin gets reduced during the light reaction. And it's very capable, ferredoxin, chemically speaking, is very capable of donating off electrons to different things going on. And in this case, to the nitrogen enzyme. Also thinking about mineral nutrition, you can see how iron is a key component, molybdenum, a key component into this enzyme. So if the plant doesn't have enough iron or molybdenum available, this enzyme is not going to get made, and you will have nitrogen fixation limited because of that. So again, just pointing out the quality of the soil, the health of the soil can make a huge difference. Okay. So some weird things about this enzyme, which aren't really being shown here in any dramatic way, but you need to know that this enzyme is very sensitive to the presence of oxygen. If free oxygen, O2, just O2 itself, free oxygen, if it is around this enzyme, it causes the enzyme to stop working. And if it keeps happening, it can fall apart. It'll just kind of denature. And so what has happened through the evolutionary process is that the microbe and the plant partner up to solve this problem. What they do, and what they've done for millions of years, is they create this molecule called leg hemoglobin. And it's essentially, when you look at what is it doing, it's binding oxygen. It's a heme protein that binds oxygen. Very similar to our hemoglobin in our bodies right now is doing that for us. Our hemoglobin's binding oxygen and delivering it to our cells so our cells can have enough oxygen to stay alive. They partner up. So they're each going to create part of this protein. So it's a multi-subunit protein again. And some of the subunits are made by the plant, some are made by the bacteria. So they both have to contribute to this. So it's a cost to both of them. It's not one being outgunned, so to speak, where they have to do all the work. And so leg hemoglobin is the name they give it to differentiate it from our hemoglobin, but it's very analogous in its function. And so when you dig up a plant like soybean, often you can see a pinkish color to the nodules. And that is because the hemoglobin protein looks pink or reddish. And so it's a dead giveaway. Sometimes when you dig up plants, you can see what looks like a nodule, but it's not a nodule. It could be, there's a disease called, for example, on soybean cyst nematode they cause these cysts. They're little tiny worms that cause cysts on the root. And if you don't know any better, you might think, oh, those are nodules. It's like, no, those are cysts. So, but if you see pink, if you see the pinkish look, then that's a sure bet. So for sake of of this class, what I'd like you to know is that the plant produces the so-called globin portion of the protein, and the plants produce the so-called heme portion. And then the subunits get assembled into the functional state of the dinitrogenase enzyme.
Is the heme portion the third portion of the complex?
Mhm. Mhm.
You
You don't need to know that, but yeah. So the heme portion is produced by the bacteria.
Oh, yeah.
Yes. And then the globin portion, so-called globin portion, is produced by the plant. And so the main goal here is let's get that made. So each partner makes their parts of it. It gets assembled into this leg hemoglobin. And then its job is to take the oxygen around the enzyme and bind it. So now it's not free. All right, it's bound on to the leg hemoglobin. And if it's bound like that, then having the oxygen bound to the leg hemoglobin doesn't hurt the dinitrogenase enzyme anymore. But there's a problem, because not all bacteria, but at least most of the bacteria that form this partnership with plants to do nitrogen fixation, most of them need oxygen to survive, just for their physiology. So it's a conundrum. It's like, okay, I need oxygen to survive, to get my carbon and energy needs met, but my enzyme can't be around free oxygen. And so this leg hemoglobin, what it does is it binds the oxygen, and then it just very slowly releases it as needed to the bacteria. Very slow, controlled release. So as soon as it gets released off the leg hemoglobin, the bacteria scarf it up for their physiology. So the dinitrogenase enzyme really doesn't see any free oxygen. So it's a pretty cool setup. Well, hemoglobin, yeah, like our hemoglobin's doing that. It's delivering oxygen. And yes, I'll show you a picture. Oh, you're jumping in. So plants will do that. And the bacteria associated with the plants, they have this leg hemoglobin strategy. That's their strategy to make sure the dinitrogenase enzyme doesn't see free oxygen, but the bacteria can stay alive because it needs oxygen. Cyanobacteria, on the other hand, they don't have that ability to do a leg hemoglobin with anybody. So they just create a special cell called a heterocyst. And it's just one of their cells in a chain of their cells that they devote to nitrogen fixation. So that cell, they keep oxygen away from it, and it just does nitrogen fixation. So there's a physical separation here where they just kind of compartmentalize where the enzyme is with respect to all of the other cells. So it's physically or structurally separate from the normal vegetative cells. And so again, that keeps the enzyme safe from oxygen. So that one heterocyst is not getting oxygen. And so again, it's just kind of fun to think about the different oxygen binding heme proteins that we have. The hemoglobin and myoglobin, and then the plant and bacteria make the leg hemoglobin. And if any of you have taken BI-218, that the protein structures here may look familiar. The helix and beta sheets and all that. And then here you have the heme, the iron chelated in the porphyrin head. So really neat strategy for the plant and the bacteria to solve that problem, to make sure that the dinitrogenase is not getting inactivated by free oxygen. Here is the heterocyst idea again, where they structurally separate it, compartmentalize it. So all these other cells are using oxygen and all of that, but this heterocyst is kept away from the free oxygen. Yeah, that at the molecular level, what it's doing, I always hear that it gets denatured is usually how they phrase it. I can double check that though to see. It might, I don't think it's a competitive binding thing. I don't think that. I think it's somehow almost like toxic in a way, if you want to think of it that way. But I'll confirm that. It's good. Yeah, from what I remember from Human Phys, yeah. Yeah, Dr. Belton, you said? Oh, yeah. Yeah. So I just wanted to alert you that when I years ago when I started doing a lot of greenhouse growing edible food in the greenhouse, I was doing research projects where we were growing like spinach in the greenhouse and soybean and on and on and on. There's a concern, especially with greenhouse grown crops, that nitrate levels can skyrocket in the tissue that people end up eating. I can't remember the exact mechanism of why greenhouse grown crops, that's a problem, but it seems to be kind of a thing. So that made a lot of people worried because it was known that if you get too much nitrate in your diet, in fact, there's laws in America that you can't have, there's threshold levels of nitrate in tissue due for food consumption. There's a law. And so there's all this concern that, you know, you want nitrogen in your diet, you want to eat food that's rich in nitrogen, but too much nitrate is considered potentially carcinogenic. So now this was published in 2018. A study came out, and one of the things it can do is cause this weird condition called methemoglobinemia, which is a mouthful. But it, excuse me, it creates a situation where our hemoglobin can't bind oxygen, which obviously is a big problem if that happens. So if it's severe enough, you can die from it. And then it's also the case where there is the potential that nitrates can be converted to nitrosamine, which are potent carcinogens, and that can get converted like that in our body. So there's been all this worry and study, and then like here in America, we limit how much nitrate. Well, then this study came out in 2018, and basically they're saying that nitrous oxide signaling actually gets boosted, which is a good thing. That's a good thing to have that happen with the intake of dietary nitrate, and it actually is helping with cardiovascular function, relating to your blood vessels and heart issues. And they say if you limit nitrate too much in your diet, it actually, at least in the mice study, led to premature death. So, here it says these beneficial effects of dietary nitrate are intriguing, particularly in light of the notoriously bad reputation nitrate has had ever since it was first proposed to promote the generation of potentially carcinogenic nitrosamine. This fear has led to very strict regulations of nitrate in our food and water. It is, however, worth noting that despite intense research by toxicologists and epidemiologists over the past decades, there's still no valid evidence for any harmful health effects of dietary derived nitrate. So I just want to put that out there, that there is, because I, for decades was told, ooh, nitrate, bad. You don't want too much nitrate. And so now they're saying, well, wait a minute, let's look at this a little bit more carefully. So I just want to put that out there for you to be aware of in your everyday life, that as with so much of science, with better studies here, in fact, a number of studies even suggest the opposite, that food naturally containing much nitrate is protective against cardiovascular and metabolic disease and even cancer. So that upturned a long-held belief I had, because I was trained that too much nitrate, bad, bad, bad. Now they're saying, wait, let's look at this more carefully. All right. Okay, so that's the end of the slide set for nitrogen, biological nitrogen fixation. I could go on and on, but we'll stop it there. Any last questions on this topic? Anything with... the leg hemoglobin? No, it's fine. No. So what happens is the oxygen inactivates the leg hemoglobin. Free oxygen. So if this enzyme is surrounded by oxygen, and I don't know the molecular mechanism, from my memory, I'm pulling out, it gets denatured, like broken down, and can't function anymore. But I don't know the exact molecular pathway. So for this class, the important thing is free oxygen, which means it's not connected to like hemoglobin. If it's just floating around free, it's bad for this enzyme.
Like hemoglobin.
I don't know. I don't know.
It kind of reminds me of like how you're saying, oh, we can like mess it up, like how when hemoglobin binds to CO2, it's stronger binding. and
Right,
Oxygen.
right. Yeah, I don't know. I don't know much about like hemoglobin other than it does a good job of controlling binding and then controlling the release of oxygen. So its job is, you know, if there's like hemoglobin around this enzyme, hopefully it's latched onto the oxygen and then it's just very slowly releasing oxygen to the bacteria so the bacteria can run its physiology. Yeah. Yeah, if that helps you link that idea, that's fine, thinking about it that way. Because if there's too much just free oxygen around this enzyme's doomed. It's doomed. All right. So it's a weird situation because so many of the bacteria that have this enzyme, they're aerobic. They need oxygen. So okay. All right, we gotta switch gears. We're out of mineral nutrition and thinking about nitrogen and nitrogen fixation. Now we're getting into photosynthesis. That's the next topic. Oh, it's not there. I swear, I'm losing my mind. I could have swore I put that up there. All right, let me go do that right now. Whoops. So let me see. All right, so we're going to break this down into like two major phases of photosynthesis. So the first one we're going to talk about is the so-called light reactions. So that's what I'm going to make visible. So now it should be visible. So light reactions of photosynthesis. So that's phase one. All right, let me reshare. Okay, so the light reactions. So now we're thinking about this amazing ability of plants to absorb light energy. So they don't need to get chemical energy. Like we have to do chemical energy. We have to eat something that was once alive or is alive. If you're eating plants, remember they're still alive as you eat them. Maybe that's not such a pleasant thought. But anyways, they don't have to do that like we do. They can just sit in the sun and that's their source of energy. They're using sun energy. So very, very different compared to what we have to do. And then what we're going to find out is that you get this electron passing process where we talk about an electron transport chain where you get electrons being passed on down the line from donor to acceptor, all the way till you finally get to a final electron or terminal electron acceptor. So it's a way to set up a situation, this absorption of light energy, it's a way to set up a situation for them to get ATP and another really important chemical called NADPH. That's the whole goal of this process. And so it sets up a situation where that can happen. And so now we're zooming into the chloroplast and we're zooming into the inside of the chloroplast. Do you remember the name of the entire membrane inside the chloroplast? Thylakoid membrane. So now we're zooming into there and we're thinking about sunlight energy. We're going to focus on leaves because most plants, it's the leaves that do the lion's share of photosynthesis. So we're going to be zooming to the leaf tissue now. We're out of the soil, out of the roots, back up into the leaves, and we're thinking about the idea of light absorption. Some fun facts here just for you to think about when you're studying. I'm not going to ask about this. It's just thinking about, you know, part of the light reactions is the production of oxygen, which we need to thank plants for, and cyanobacteria because they're providing that oxygen that allowed for us to evolve on this planet. So this is just some interesting data to think about. So it depends on where the plant is situated, how much oxygen it releases. The desert is a much slower, lower process. Okay. So now we got to think a little bit about light. And if you've had physics, you've probably thought about light already, but let's think about it if you've not had physics. So we're thinking about this radiant energy. And the idea of an electromagnetic spectrum coming from the sun. When light is traveling from the sun to the earth, as it travels through the atmosphere, it behaves like a wave of energy. Okay, so we talk about the wavelength of that energy. And for us, we see in the visible spectrum, so we can detect wavelengths of light in the 400 to 700 nanometers. So we usually talk about the different wavelengths and then we call them different nanometers, right? That's the unit we use. So 400 to 700 is visible light that our retina, our eyes can see. But there's other wavelengths below and above that that we can't perceive, but other organisms can. Honeybees are famous for being able to see in other qualities of light that we can't pick up. So through the atmosphere, it behaves like a wave. When it is interacting with matter, like chlorophyll, it behaves like discrete particles. So Einstein was very famous for better, helping us better understand this process through the research that he did, that it has both of these properties. It acts like a wave through the atmosphere and acts as discrete particles when it interacts with something physical like chlorophyll. So here you're seeing just the wavelength idea that you have this wavelength of light quality, and the wavelength can vary, be very long, it can be very short, and that would correspond to a different color of light, a different quality of light. And so here you're seeing the visible spectrum in the 400 to 700 range that our eyes can pick up on. Okay, so that's very human centric. It's saying what humans can pick up on. But honeybees, when they look at a flower, if you've taken Plant Kingdom, we show you pictures of what we see when we look at a flower, the different colors, and then they do special imagery where they show what a honeybee sees when he looks at that flower. It's very, very different because they can see into the ultraviolet. And so they can pick up ultraviolet, and it can be very dramatically different. So you can see that there's other wavelengths outside of our visible spectrum, the infrared, microwave, radio wave. And of course, you get into ultraviolet, X-ray, gamma rays. These are very energetic. They have very small wavelengths, but very high energy, right? So they can be damaging. Too much of that can be damaging. All right. So we're most interested for this discussion. We're thinking about what happens when light interacts with the chlorophyll pigment, and the idea that it's interacting with a physical, chlorophyll molecule. And so we start talking about photons of light energy that are interacting with the chlorophyll. And the energy that's carried by that photon is called a quantum. If you ever hear or see that term, that's referring to the energy. The energy, the quantum energy, EQ is quantum energy, is equal to hc divided by lambda. And let me, so c divided by lambda, you have the frequency taking the place of that. So do I have that? Oh, it's in the notes. So frequency is related to the wavelength, and that's v, that's the v. And it's the c is the speed of light, h is Planck's constant, c is the speed of light, and lambda is the wavelength. So the c divided by lambda term in this equation, that's the definition of frequency, which is that fancy italic v. Okay, so I'm not going to ask you to memorize this per se, but some people like to think about, I usually think about it more in this way. I don't know why, but I like to fiddle with the wavelengths, and sometimes that helps me figure out what's going on in terms of what quality of light is going to be giving you more quantum energy. I'm not going to ask you to do any calculations. It's just if your mind works that way, where you like to play with equations as you're studying to try to tease things apart, you're certainly welcome to do that. Okay, so the matter that this photon is interacting with, we're interested in the chlorophyll for this class, and thinking about that pigment and how it can absorb this quantum energy. So what we see happening when we look at the chlorophyll pigment, when it absorbs light energy, it's exciting electrons of that pigment. So we're zooming in to electrons of chlorophyll. And when it absorbs quantum energy, that light energy, it will excite that electron in the chlorophyll. And that causes the electron to get elevated to a higher level, a higher energy level. And then if it's at the right excitation level, it can do something. That electron can do something that can be very useful for the light reactions. But there's options. There's like several things that can happen to that electron. And so we refer to that as the excited state. So when we talk about that electron being excited, that means that that chlorophyll has absorbed light energy. And it has led to this excitation state of the electron of the chlorophyll. Here, this is just showing the sunlight energy spectrum and putting it into context of what the chlorophyll likes to absorb, what qualities of light it likes to absorb. And so the green is what we see here for chlorophyll. So when you extract chlorophyll and you just put it into a spectrophotometer and then you run a spectrum on it, you say, I want this pigment to go through a 400 to 700 wavelength absorption and see what happens. And what you would see is this data would be spit out, where you see this peak and then drop down, not much absorbance, and then another huge peak of absorbance. And those correspond, they have the visible spectrum down here to show you what qualities of light chlorophyll has evolved to maximally absorb, and it's red and blue. All right, so chlorophyll loves red and blue light. It's really well adapted to snag those qualities of light coming from the sun, whereas it doesn't care very much about all the other qualities of light. So red and blue is what it's evolved to maximally absorb, which that's a good thing. If it's maximally absorbing, that means it can really do something with that energy now. It's good. All right, so the chlorophyll is really using only a couple of options coming from the sun, red and blue primarily, okay? All right, so we have to imagine that chlorophyll absorbing sunlight energy. It preferentially absorbs red and blue. By the way, that's why, leaves look green to us, because it won't, chlorophyll doesn't like green quality of light, so that just bounces back to our eyes. And that's why, leaves look so green. And then some of the other qualities of light can pass through the leaf into the lower canopy in a forest system. So they can bounce back, they can let some pass through, but they really, really like that red and blue. Hey, that rhymed. So excess energy can be dissipated. So let's, the chlorophyll absorbs that light energy, that electron gets excited. So now that electron's all excited, and it's like, okay, it could just dissipate. If things aren't perfect, it could just dissipate, which when we say that, usually what we mean is that it drops back down out of its excited state. And as it does that, it releases some heat energy. Okay, so heat energy gets released. It might get excited and then drop to a lesser excited state that causes a fluorescence phenomenon. And they've done lots of cool experiments with that, where they can make the chlorophyll fluoresce, and they've perfected that. So that could happen, where it drops down to a little bit lower energy, not all the way down to the ground state, but a little bit lower, and that will give a burst of light that's of a longer wavelength. So that's not really what we're going for when we think about the light reactions. Neither of these. It's like, that's not ideal. You don't want this going on for the light reactions. Or that excited electron could get transferred. Well, no. Let me back that up. The energy of that excited electron could get transferred, not the electron itself. Here I'm just talking about the energy. This is a weird concept. The best way, this concept is called inductive resonance, or energy transfer. And this is really it's always been challenging to wrap my head around. It took me a while to get comfortable with it. The best analogy I can give you is if you've taken music class where you play around with tuning forks, and you hit a tuning fork and it starts to give off, a musical note, and then you pull it closer to a tuning fork that's just still, you didn't hit it, and that tuning fork starts to hum at the same frequency, so you get the same note. That's inductive resonance. You have not physically touched them. There's no electron jumping from that tuning fork to this tuning fork, and yet you got energy transfer to allow that tuning fork to start to hum with the same frequency as the one you hit and is humming along. Do you remember doing that as a kid? Tuning fork? I remember like elementary school. It was just like, that's freaky. It was really freaky. Tubes? Oh yeah. Yeah, I've seen those too. And like science museums, they're little science things you can buy. So that's inductive resonance. I do need you to know that term because I'll be talking about that term. So it's energy transfers. It's weird. I know we got to give that some thought. And then sometimes that electron, and hopefully this is going to happen too. So we want energy transfer. That's a good thing, actually. So during the light reaction, inductive resonance, we want some of that going on. I'll explain where. And then we also want this last one, the reverting to an excited triplet state. And I'm not going to get into the physics of that. This is not a physics class, but the triplet state is stable enough. It's still excited, but it's stable enough that it can stick around in that state long enough for electron transfer to happen. Not just energy transfer, but electron transfer. So the triplet state is good for once it gets to that excited state, it's like, okay, I can play now. I can give off this electron. So it's more stable, it sticks around longer in that excited state. And if everything goes okay, it's going to lead to a photochemical reaction. When we say photochemical, that's when we mean an electron from that chlorophyll, that excited electron, is going to get passed off to an acceptor. That's a photochemical reaction. Okay, so now if that chlorophyll gets excited by light energy, that electron gets boosted to the triplet state. If it gets donated off to an acceptor, now that chlorophyll has lost an electron. So then we say it's photooxidized because an electron's been taken off of it, given to some acceptor. Now it's missing an electron. So the pigment is going to want, so to speak, to get an electron back and not keep missing these electrons. That would not be good. So what has to happen is there needs to be a source of electrons to fill that hole, so to speak. So the chlorophyll electron gets excited, donates off to an acceptor, and now the chlorophyll is going to go, okay, I need an electron to fill that now. And so there has to be a source, a pool, where the plant can say, okay, here's an electron to fill that one you just lost. And that has to keep happening in picosecond timing to keep this going all day long. And so once that happens, once the chlorophyll says, hey, I need an electron, and then that electron's given back to chlorophyll, and now it's got its full complement of electrons. Now light energy can strike it again, and that can happen all over again. All right, so it's not like the chlorophyll can just give off all of its electrons one by one by one. That would destroy the pigment, and that doesn't make sense. So there needs to be a constant supply of electrons to fill the gap or the hole left by the ones that are given off to the acceptor molecule. So this is a visual showing some options of what can happen when chlorophyll gets excited. Here's your ground state where there's no excitation, down on the bottom x-axis, so to speak. And then here you're seeing on the y-axis some options of when the chlorophyll gets hit with different wavelengths of light. So you can see here, this is colored blue. So they're saying if the chlorophyll absorbs blue light, and then compare it to if the chlorophyll absorbs red light, which one excites the electron more? What quality of light? Blue. You see it go up on the energy scale here on the y-axis. Higher it up, the higher the energy. So blue is a shorter wavelength than red. Yep. So higher energy, higher frequency. So here it's actually showing that when a plant absorbs blue light, you actually get a higher excitation of the chlorophyll electron. Here they're showing that maybe it'll drop down part way, the electron will drop down a little bit and you get some heat loss. There's fluorescence, phosphorescence, we're not going to get into that. the main thing on here is that you notice that there is a difference in excitation depending on what quality of light, and it does matter. And then here, T, right there, that's the triplet state. So you can see it's dropped down quite a bit, but it's still excited. It's not at the ground state. This is the more stable, excited state, the triplet state. So now, once the electron gets to that point, it can do something. Hopefully it'll give it off to an acceptor. Here on the figure B, they're just mapping out the wavelengths of light that are maximally absorbed by the chlorophyll, and you can see it corresponds to the red and blue. Okay.
So even though it's like going to a higher excited state, does it more special?
Yeah, I mean, basically they still need to get to the triplet state, you know.
So does it matter?
Well,
Or does it doubt?
I mean, I think if you play with the different qualities of light, you'll see the plant. There'll be differences. Most grow lights, I think, are a combination of red and blue. Although, yeah, they look purplish. But yeah, you can get the lights that you can play with. Like I just want red light or I just want blue light. Those are always fun to play with and see what happens. And you should see shifts in the growth. They've been publishing some nice, carefully controlled studies on LED lights and the different qualities of light. And there's some really interesting impact just even on, like the secondary metabolite production. You can make the plant make different secondary metabolites or more of a certain secondary metabolite just by shifting the light wavelengths you expose the plant to. So anthocyanin production, yeah, there's been pigment production. I've seen other secondary metabolites. So a lot of the indoor ag and controlled environment ag, this idea of, it's exciting to think that you can manipulate the plant's environment in a very fine-tuned way to direct the plant's physiology to what you want. And so it's the potential there is good for if you want a certain product, and if you can figure out the way to manipulate the environment, you can increase your yield quite a bit and your profit margin. So and then, the edible food idea, healthier food, anthocyanins are very good for us. So upping anthocyanin production in the plant, for example, is good for our nutrition. So they're looking at all of this. How do you manipulate in controlled environments? And light is a great way to do it. Simple, it's fairly cheap, fairly easy to manipulate. And so they're looking intensely at that in the scientific research. So basically this light is acting, or this pigment is acting as a photoreceptor is what they call it. And it's, photoreceptors are essentially pigments that absorb light. There's more than just chlorophyll as a photoreceptor. We'll talk about photoreceptors a little bit later, other types. Chlorophyll A is one of the major ones in the plants, the typical plant that you think about. And the chlorophyll A structure is kind of a two-part structure. It has a porphyrin head region, and then it has the so-called phytol tail region that anchors into the thylakoid membrane. So that's a really good portion. The phytol tail is got a lot of the chemistry very similar, that it can interact with the membrane of the thylakoid very well and dock into it. Don't forget, magnesium is chelated in the center of the porphyrin head, so plants need a lot of magnesium.
Do you think chlorophyll A is the most prominent, like chlorophyll?
Chlorophyll A, and then let me point out chlorophyll B. So in the typical plant that we think about, chlorophyll A and chlorophyll B, the only difference between those two is this methyl group is replaced by an aldehyde. So chlorophyll A and chlorophyll B are super dominant or predominant in like typical plants we grow for human use. But there's other pigments. Bacteria chlorophyll is similar to chlorophyll, but it's found in bacteria. We'll talk about carotenoids. So here are carotenoids. So we'll look at these. Yep, exactly. So we'll look at these again on Wednesday and start getting into all this electron gain for the light reaction.